Today we are going to see the physiology of cerebellum. Okay, the presentation will follow this order. We'll see a little introduction, the functional anatomy of cerebellum, the different divisions of cerebellum, the histological structure, the neural circuits and neuronal activity, the functions of cerebellum, the cerebellar lesions and signs of cerebellar dysfunction, and the clinical tests. So cerebellum is derived from the Latin word meaning little brain. The cerebellum is the largest part of the hindbrain and it is located in the posterior fossa of the cranium just below the occipital lobe. Okay, it weighs about 150 grams. It is also called as the silent area of brain because the electrical excitation of the cerebellum does not cause any conscious sensation or rarely causes motor activity. Okay, in its absence or when the cerebellum is damaged, the movements of the body becomes highly abnormal. That is why it is termed as the silent area of brain. Now coming on to the functional anatomy, the cerebellum is derived from the metencephalon of Roman cephalon. It is made up of a vermis, the central part, vermis, and lateral to the central part we have the hemispheres called as cerebellar hemispheres. The cerebellum is connected to the cortex, the brainstem and the spinal cord through the superior, middle and inferior cerebellar peduncles. So this is a mid-sagittal section of the cerebellum. Here you can see the cerebellum. There are numerous folds, right? Transverse folds. These are called as folia. And this is the white matter and you have a outer grey matter too. So moving on to the different divisions of cerebellum, we have anatomical division, phylogenetic division and functional division. Anatomically, the cerebellum is divided into anterior lobe, posterior lobe, flocculonodular lobe. And phylogenetic, it is based on the evolutionary process. It is divided into archicerebellum, paleocerebellum, neocerebellum. And depending upon the functions, vestibulocerebellum, spinocerebellum, corticocerebellum. The anatomical divisions, the cerebellum has two surfaces, that is the superior surface and inferior surface. The superior surface is related to the tentorium cerebellae and inferior surface to the hollow lobe of the occipital lobe. Okay. And the cerebellum is divided into three lobes by two fissures. It has three fissures, namely the primary fissure, the horizontal fissure somewhere here, and the flocculonostrolateral fissure. And three lobes, the anterior, posterior and flocculonodular lobes. So these are the fissures of the cerebellum. Here you can see it's a primary fissure, the horizontal fissure, the posterolateral fissure. Okay, and middle part is the vermis, and lateral to the vermis you have the hemisphere, that is the cerebral hemisphere. So this is a vermis. Vermis is nothing but a narrow band-like structure which separates from the cerebellum. It has related to the vermis is the cerebellar hemisphere. It is further divided into two. One is the intermediate zone, this part that is immediately lateral to the vermis, intermediate zone, and lateral part of the cerebellum is called the lateral zone of the hemisphere. Okay, so this vermis is worm-like body, and its main function is to control the activity of your head, body, neck, hip region, shoulder region. Whereas the intermediate controls the distal portions of your fingers, toes, feet and all. And the lateral zone is responsible for the overall motor activity, that is planning and how it has to proceed. In that way, the lateral zone of hemisphere is concerned. And moving on to fissure, this is the primary fissure. You can see it is present in the anteroposterior aspect of the cerebellum. Okay, it is V-shaped in nature. Okay, it is present in the it forms the posterior limit of the anterior lobe. The horizontal fissure, you can see this is the horizontal fissure. It divides the cerebellum into two surfaces, that is superior and inferior. It has no functional significance. Whereas a posterior fissure, it divides the it is in this region. So you can see the posterior lobe and flocculonodular lobe lies between them. Okay. So the primary fissure in this, you can see it is a V-shaped. Can you see V-shaped? Okay. Horizontal fissure, it is see it is it divides the cerebellum into superior and inferior surface, and it has no functional significance. The posterolateral fissure divides the posterior lobe and the flocculonodular lobe. Okay, the lobes of the fissure, you have the anterior, posterior and flocculonodular lobe. The anterior lobe is the part of the cerebellum which lies in front of the primary fissure. The posterior lobe lies between the primary fissure and the posterolateral fissure. It has both the surfaces of the cerebellum, the superior as well as the inferior surface of the cerebellum. The flocculonodular lobe is a part of the cerebellum that is anterior to the posterolateral fissure. It has a flocculus and nodule. The flocculus is the nodule is nothing but the rostral part of the vermis, whereas the flocculus is a irregular shaped grey mass. Moving on to the phylogenetic division, based on the evolutionary process, the cerebellum is divided into archicerebellum, paleocerebellum and neocerebellum. The archicerebellum is the oldest part and it involves a flocculonodular lobe and lingula. The paleocerebellum is next to appear and it involves the anterior lobe except lingula and the following parts of the posterior lobe. And neocerebellum, it is the latest part to develop. Moving on to the functional division, we have vestibulocerebellum, spinocerebellum, corticocerebellum. Vestibulocerebellum, as the name suggests, it has connections from the vestibular part. Okay, so it is mainly responsible for the control of body posture and equilibrium. The spinocerebellum, it is mainly for the proprioceptive inputs from the spinal cord and also controls the axial and limb muscle reflexes. The corticocerebellum is also called central cerebellum. It occupies the lateral regions of the cerebellum and it is mainly responsible for the smooth performance of the voluntary movements. So this is the vestibulocerebellum, spinocerebellum, cerebrocerebellum part. Okay. Now moving on to the histological structures. Histologically, the cerebellum has three layers. One is the cerebral cortex, which forms the outer grey matter, and white matter, which is formed by both the afferent and efferent nerve fibers. So it's like it has both the input and output. Okay, input to the cerebellum and output from the cerebellum. And deeper to that, we have the deep cerebellar nuclei. So the outer cortex layer is divided into three, that is the outer molecular layer, middle Purkinje layer and inner granular layer. The outer, it has some neurons, and the molecular cell, it has the stellate cell and basket cell, and the Purkinje cell layer we have the Purkinje cells, and the granular layer we have the granule cells, Golgi cells. So this is another picture showing the outer cortex, cerebellar cortex, molecular, Purkinje and granular. Here you can see in molecular layer you have the stellate cell and basket cell. Purkinje layer you have the Purkinje cells, and granular layer you have the granule cells and Golgi cells. Apart from this, you also have some fibers. This is mossy fibers and these are the climbing fibers and parallel fibers. The molecular layer you can remember as the outer cortex cerebellar layer, you can remember as my personal garden. M in my represents the molecular layer, P in personal represents the Purkinje layer, and G in garden represents the granular layer. So moving on to the molecular layer, you can see it has stellate cells and basket cells. The stellate cells synapse with the parallel fibers of the granule cells and also synapse with the dendrites. That means the stellate cells axons, okay, synapse with the dendrites of the Purkinje cells, whereas the dendrites of the stellate cells synapse with the parallel fibers of the granule cell. And also the basket cells, which gets input from the parallel fibers of granule cells, and this synapses with the cell bodies of the Purkinje cells. I'll show you. See, in this diagram you can see the stellate cells, the dendrite synapse with the parallel fibers, whereas the axon synapse with the dendrites of the Purkinje cells. This is basket cells. It receives input from the parallel fibers and synapse with the cell bodies of the Purkinje cells. Okay. So the basket cells can synapse with about 70 Purkinje cells. So that's why it has got the name basket cell. And apart from these two cells, the molecular cell layer also has some nerve fibers. Okay, these are the parallel fibers, the dendrites of Purkinje cells, and climbing fibers from the inferior olivary nucleus. The Purkinje cell, which is the biggest neuron in the body, the Purkinje cell, dendrites of the Purkinje cell extend into the molecular layer and forms axodendritic synapse, and they go deep to the deep cerebellar nuclei. They are the only output neuron from the cerebellar cortex, and they are inhibitory also. Okay. So these are single layer of large flash shaped cells, biggest neuron. It is a only output neuron and exerts inhibitory influence to the deep cerebellar nuclei as well as to the lateral vestibular nuclei. The granule cell layer has two cells. One is a granule cell and Golgi cell. The granule cells are very small, numerous. The axons of the granule cell ascend into the molecular layers and form the parallel fibers that are excitatory with and form synapses with the Purkinje cells, stellate cell, basket cells and Golgi cells. The Golgi cells are less numerous compared to the granule cell. Their dendrites project into the molecular layer and receive input from the parallel fibers and also from the climbing and Purkinje fibers. Their axons sends inhibitory impulses to the dendrites of the granule cell. Okay. The sensory inputs, we have the climbing fibers and the mossy fibers, which are both excitatory in nature. So this is a histological picture. Here you can see this is the mossy fibers and this is the climbing fibers. The climbing fibers are from the inferior olivary nucleus of medulla. They travel via the olivocerebellar tract to the cerebellum, and it has one-to-one connection with the Purkinje cells. So it's like one climbing fiber for 5 to 10 Purkinje cells. The climbing fibers produce action potential for prolonged period, that is up to 1 second. So the action potential produced by this climbing fibers is known as complex spike action potential. The mossy fibers as its origin from the spinocerebellar tract, reticulocerebellar tract, vestibulocerebellar, cuneocerebellar tract, corticopontocerebellar tract, and they synapse with the dendrites of the granule cell and form synaptic glomeruli. This synaptic glomeruli exerts an inhibitory effect from the Golgi cells. The action potential of the mossy fiber is not as like of the climbing fibers, and it is called as simple spike action potential. Of the 4 cells, the only excitatory is the granular cell, and it releases the neurotransmitter glutamate. And all the 4 cells are inhibitory in nature, and it releases the neurotransmitter GABA, which is also known as gamma aminobutyric acid. That's all about the outer cerebral cortex. Okay, outer cerebellar cortex, we have the molecular, Purkinje and inner granular layer. The outer molecular layer has stellate and basket cells. The middle Purkinje as Purkinje cell, and the inner granular as granular and Golgi cells. Apart from this, we have the sensory inputs like climbing fibers and mossy fibers also. Okay. Now moving on to the white matter of cerebellum. The white matter of cerebellum is formed by both the afferent and efferent fibers. These fibers are classified into three groups. One is the projection fibers and another one is the inferior association fibers, commercial fibers. The projection fibers, as the name indicates, they leave or enter the cerebellum to other parts of the central nervous system. There are three types. One is the inferior cerebellar peduncle, which connects the cerebellum with medulla. It has input to the cerebellum. The middle cerebellar peduncle, which connects the cerebellum with pons, it is also input to the cerebellum. The superior cerebellar peduncle that connects the cerebellum with midbrain. Okay. The association fibers, these connect the different regions of the same cerebellar hemisphere. The commercial fibers connect the areas of two halves of the cerebellar cortex with each other. So the cerebellar peduncle, superior cerebellar peduncle, also known as brachium conjunctivum, which is the output from cerebellum. The middle cerebellar peduncle, which is the brachium pontus, it is input to cerebellum. The inferior cerebellar peduncle, restiform body, input to cerebellum. Now moving on to the deep cerebellar nucleus. Okay, we have four pairs of gray masses that are located on the roof of the lateral fourth ventricle. Okay, so these are also known as roof nuclei. We have four pairs of gray masses. Okay, one is the dentate nucleus, emboliform nucleus, globus nucleus, fastigial nucleus. The dentate nucleus, you can see it is the largest of all the four and it is purse-like shape. It is crenated lamina and open in the anteromedian region. Through this region only the superior cerebellar peduncle fibers connect. Okay. And just medial to it you have the emboliform nucleus, which is oval mass of gray matter. And just medial to the nucleus emboliforms is another nucleus called as the globus nucleus. These together called as, this emboliform and globus together called as nucleus interboiscus. And the nucleus fastigius lies nearly in the midline and is spherical in shape. Okay, it lies just over the roof of the fourth ventricle.